Morning, guys. Dr. Gillard here. Let's do some embryology. Brand new section. I just made this over the last few days. Probably one of the most complicated stories I've put together thus far. So if you're watching this on YouTube, check back because I'll probably, maybe in a couple quarters, I'll add another version of this as I get smooth it out a little bit. But warning, great author confusion. What a disaster. Authors are all over the place on this thing. Complete disaster. Probably the worst author mess up. I don't want to say mess up, but man, they just can't agree on the steps, and it's a mess. Also, if you are not familiar with meiosis, I know everybody's had that in high school these days, but I would go watch the Amoeba Sisters. If I can remember, I'll put a link down below, but just go on YouTube and click Amoeba Sisters meiosis, and they have wonderful videos. They're made really simple, but they're very accurate, and they will definitely help you to understand, especially to understand the difference between meiosis 1 and meiosis 2. Some people call it meiosis 1 and meiosis 2, tomatoes. So you get more out of this if you do that first. Okay, so we'll talk about oogenesis, pronounced O-gen-e, like egg, cis, oogenesis, we'll be talking about. And that's the process of forming an egg. Last class, we talked about how to form a sperm. Pretty straightforward, really. Compared to this, it's pretty easy to make a sperm. Oocyte is not an easy task, as we will see. Star of the show here is kind of the ovaries. Okay, so it all starts out, I'm not going to go crazy in this because I'm going to cover this later in the course, more specifically. But for now, the tissue destined to become the egg, in other words, the primordial germ cell of the egg, they actually start out as yolk sac tissue, which slowly crawls out of the yolk sac, and it gathers on the gut tube, which we will get to in this class. It's one of the last lectures I do, but you'll see what the gut tube is. And there, it induces some surrounding tissue to surround it, to kind of support it, so it gets some supporting tissue. And the supporting tissue grows through mitosis, not meiosis. Mitosis to form these bumps. And these bumps, which are on the gut tube, are called the genital ridges. These will be pulled down into the ovary position later, and we'll talk about that probably in 5th quarter. But anyway, these bumps are what's going to give rise to the ovaries, and inside the ovaries are the primordial germ cells that will ultimately form an egg. And remember that all these germ cells are 2N, just like all germ cells are. They're 2N. When I say 2N, that means they're diploid. They have 46 chromosomes, and yeah, they are going to divide and divide and divide and divide and divide until thousands or hundreds of thousands of these primordial eggs are created, all of which, because we're dividing through mitosis, are 2N 46 chromosomes. Then they're eventually going to turn into primitive eggs, which are called oogonia. Oogonia. That's plural. Singular is oogonium. Authors surprisingly screw that up as well. They go through the whole book saying oogonia, never once oogonium, and they're using this as singular and plural, and that's not the way to do it. Anyway, 3rd month. So the 3rd month of development, still in your mommy's tummy, just a little tiny thing, right? The 3rd month. The oogonia begin meiotic cell division. And okay, great. So they go through interphase, no problem. And they start into, remember IPMAT? They go into interphase, and they have a problem. They make it through the crossover stage, but then all of a sudden they stop. And they're stuck in prophase. Specifically, Carlson, which is my favorite. That's kind of my closer, though. I don't build a lecture series looking at Carlson because he's so deep. Larsen did a really good job of telling this story and getting things going, better than all the other books, even better than Carlson. But then when you come in with Carlson at the end, it fills in all these gaps. So that's a great way to build embryology lectures. But anyway, where it really stops is called the diplotene stage of prophase. Remember, prophase has several stages within it. But I think that's probably, for chiropractic boards, probably too deep. Medical boards, you probably should know those other stages. Anyway, at this point, when it stops going through mitosis and stuck in prophase 1, you are said to be, if you're a cell, you're said to be in meiotic arrest, meaning you're stopped, cardiac arrest, your heart stops. Meiotic arrest, the meiosis is stopped, and you're stuck in prophase 1. Nevertheless, once those primordial germ cells start meiosis, even though they're stuck in prophase 1, they get a new name, and they're called primary oocytes. Primary oocytes. Definitely want to star that. I didn't have time to put many stars because this is my first lecture on this. What stops meiosis? Okay, we got to dig into Carlson to see. None of the other books go this deep, but probably for medical students, you should know this. Or if you're chiropractors, I'm not sure if you need to go this deep. Maybe the top tier questions, some might be about this. So I could ask this as well, but you know how that goes. Anyway, so meiotic rest occurs when the cells outside of the oocyte pump 2 molecules into the cytosol. So remember, those support cells are still around there, and we think that those guys make cyclic AMP and GMP. I think GMP is actually made, cyclic AMP technically is made in the nucleus. Cyclic GMP is pumped in there. So we'll look at gap junctions that these things are pumped for. And this is important. We need to block those gap junctions later on to wake this up again, as we'll see. So cyclic AMP and cyclic GMP are pumped into the cytosol. They get into the nucleus and they freeze. They cause meiotic arrest, and the cell freezes in prophase 1. Okay, and mechanisms too complicate us. These two shut down meiosis. We won't get into all the details. If you're a biochemist in this stuff, PhD, Carlson goes quite a bit deeper, so that's a good book again to get for that. So only prior to ovulation, just before ovulation, we're going to wake prophase 1 up and go into meiosis 2. But we got a ways. We got a lot to talk about before that happens. All right, so prior to ovulation, with the concentrations of these will decline in the nucleus of the champion oocyte. What the heck is the champion oocyte? We'll talk about that. So these primary oocytes, these guys wait a long time. Some of them can wake up at 12 years of age or so when menses starts or when menarche starts. But some of them may not wake up till you're 30, 40, or even 50. So some of these primary oocytes are very small, right? They're almost microscopic, live in your ovary, and they hang out there for decades and decades. Some of them are lucky and get woken up much sooner. How can they wait a long time? What about the damage if they wait so long? Well, there is. That's why people over 40 have a greater chance of developing birth defects. Because these primary oocytes have been sitting on there. They've been x-rayed. They've been flying airplanes back and forth, which exposes you to some radiation. Yeah, there's a lot greater chance of damage. So the nucleus of these, what protects them? So the nucleus of these meiotically arrested primary oocytes begins to accumulate a bunch of fluid. And we'll just leave it at that. That fluid gives some extra protection to the chromosome from environmental damage. We'll just leave it at that for here. Again, if you want to go deeper, go into Carlson. So around the 4th month of development, something changes. Now, this is arguably, authors are all over the place on when all this stuff happens, but I'm trying to stay with the board books for the most part. So around the 4th month of development, a new single layer of squamous cells shows around the primary oocyte. It encircles the oocyte. They think it's a drive from some of those ovarian cells that are in the vicinity. Not 100% sure. So we have a single layer of simple squamous, and we know simple squamous, right? They're flat cells. Those are simple squamous cells. So there's a bunch of them that surround the oocyte in the middle. There's the oocyte. I think I have a picture. I don't need to draw this. But those are squamous cells. And yeah, so they have a basal lamina that sits outside of the oocyte. So if this is the oocyte here, and we got a new layer that has shown a pair of squamous cells, okay, those are called follicular cells. There's a little cell in each one of these. Follicular cells make up the follicular epithelium. It's a single layer, but these cells, are flat. I didn't draw that very good. And they sit, I can change colors here, they sit on a basal lamina, which is a little bit weird. Usually I guess that's the way a blood vessel works. So this is the oocyte here, the primary oocyte. And so the basal lamina is deep to the nuclear membrane. There's the nuclear membrane of the oocyte right there. And it's deep to that layer is what I'm trying to say. So anyway, I'll show you a nice picture in a second. So this new follicular epithelium, single layers, squamous cells, plus the primary oocyte that it wraps around, and the basal lamina, or basement membrane, they are collectively called the primordial follicle. Primordial means before birth, for the most part. Primordial follicle. So this follicle, hard to see. You need a microscope to see this thing. Really super small. But yeah, it's there. Just remember, make a note card. Primordial follicle. Before birth begins, these are found. They're the follicular epithelium plus the primary oocyte is the primordial follicle. Everybody good with that? Okay, these here's a kind of a statistics page. These primordial follicles are really delicate, and that's all mammals go through this. Zillions of primordial follicles, not zillions, but millions of primordial follicles. 7 million start out in the 5th month within the ovaries. That's a lot of primordial follicles, but they're delicate. They degenerate away, in fact, by birth, we've lost over half of those things, right? There's only 2 million left by birth. So we've lost a ton of those things. And by the time menarche begins, make sure you know that if you don't know. Menarche is puberty. That's when you start your menstrual cycle. So that's usually around the age of 12 or so. But there's only 40,000 left. So we started with 7 million. And we dropped all the way down to 40,000. All right. And of the 40,000, only 400 will ever get to become one of the big 3 secondary oocytes. We'll talk about that whole process in a minute. Carlson says that 99% of all these primordial follicles that contain the germ cells, the primary oocytes, they're lost via apoptosis, cell death, during a woman's lifetime. Most of the death occurs prenatally before the baby is born. So bottom line, these things are super delicate. All right, so what happens next? We have a primordial follicle. Our baby is just born. So for the first 12 years, for the most part, these primordial follicles just hang out and do nothing. So all 40,000 of them, they are dormant. However, once menarche gets close and starts, and menarche is the key of that is the anterior pituitary releases follicle stimulating hormone, FSH, and luteinizing hormone, LH. Once those two start flowing a little bit, things are going to change. In fact, when menarche starts, a lucky few of these are actually awoken, and not all of them, just a tiny, like 50 of them at the most. They're woken up, and then one of them, usually one of them, sometimes two, but usually one of them has a chance to escape the ovary prison. The rest of these 700 or 7 million primordial follicles with a primary oocyte inside, those primary oocytes never leave the ovary. So it's very rare to get a chance to get out. But a few of them get out, and then those few are going to race to see who can develop first. One of them will be the winner. 3 of them will make it to the finals, but only one will be the winner. What wakes up the primordial follicle? They used to think, back in the day when I went to school, they thought it was pure FSH and LH being secreted, but that's not exactly true. We're not sure what initiates the starting group. About 50 of them, anywhere 5 to 50. We'll go with Carlson says 50. Larsen said, I think, 5 to 15. So they're all over. Every author says something different. We'll go with Carlson because he's the most evidence-based. But anyway, quite a few of these tiny primordial follicles begin the race. We're not 100% sure what wakes them up, because they wake up and you do testing to see if LSH and FSH hormone levels are in these kids' blood, and they're not yet. So we don't know what starts them off. Those hormones are super important for running the race and growing big, but it doesn't wake them up, so we're not 100% sure what initiates them to grow. And as I said, of this group of 50, only 3 of them will make it to the finals, where they become quite big, 8 millimeters in size, according to Carlson. Right? The rest of them die. We'll actually see how some of them are killed. Once they're formed and start the race, though, hormones are the key. Hormones drive the growth of follicular cells, as we'll see in a minute. Follicular cells are what makes the mass of the follicle get bigger. So once we wake up from menarche, primordial follicles, they need hormones to start the race. Hormones come from the HP axis, the hypothalamus and the pituitary. And we'll get into this more in 5th quarter. And you'll probably have this in someone else teaches some endocrinology, I can't remember who, but you'll have this. Gonadotropin releasing hormone is released from the hypothalamus. That goes down and stimulates the pituitary to release the gonadotropins. The gonadotropins is a word that collectively means follicle stimulating hormone, FSH, or luteinizing hormone. So gonadotropins, if you want to talk about these two without saying FSH and LSH, you can just say gonadotropins, and it's understood we're talking about FSH and LH. Right? Everybody good with that? And we'll see how mainly this is the main one that drives growth and development of the follicle, because we have a microscopic follicle, and that thing's going to get up to 20 millimeters, almost an inch. It gets so big you can see it with a naked eye easily. So FSH is the one who drives most of this, although LH is important as well. I'm not going to get super deep in that until 5th quarter. Follicular cells, growth and enlargement. Follicular cells, so remember we had those, so far all we know is we have squamous cells, right? We have a bunch of squamous cells. I'll draw them again. I thought I had a picture in here somewhere, but it's coming. So we have these flattened single layer of cells. This is the follicular epithelium, and then we have the oocyte stuck in prophase 1. So these are also called granulosa cells. So those are the fasciculus cells, also called granulosa cells. So they have FSH receptors, right? Let's make them, how about pink? They have FSH receptors here. And so here's FSH. They can bind FSH receptors, and through mechanisms we won't explain, that stimulates them to multiply. And they multiply, and they get thicker and thicker, and this layer gets thicker and thicker. But that's how this thing works. But FSH receptors are super important, and they're on those cells. We're going to morph these into another. Before we grow them, though, I probably shouldn't have put that in there yet, because we got one step we have to talk about before that happens. So the binding of granulosa cells, that stimulates the production also of estrogen, That estrogen also helps in the preparation of the uterine wall, as well as help of growth and development with the follicular cells as well. I'm not going to go down that estrogen rabbit hole right now, though. All right, so we had a primordial follicle, and now let's meet a new follicle, a primary follicle. Now, this is arguably, because some authors actually think the primary follicle is created before birth. Some think it's created after birth. So go with board books here. So the first thing that happens after birth, and now these are not of all the cells. The primordial follicles are still waiting and waiting. Remember, only 50 of them woke up. So now we're talking about 50 of them are in a growing race. And so this is what I'm talking about. I'm not talking about the rest of the primordial follicles. So the first thing that happens is that single layer of epithelial cells, it morphs into a new layer, morphs into a cuboidal layer. And at that point, once the cuboidal cells replace the squamous cells, we get a new name. Again, this is arguably, we get a new name. For our purposes, primary follicle is born. So you no longer have primordial follicle, it switches to a primary or a growing follicle. I shouldn't call it that because there's actually a name for that coming. But when you see, for our purposes, when you see primary follicles, that means that this is one of the 50 cells that's been selected for the race, and that inner membrane has changed into cuboidal instead of squamous cells. So the follicular cells or follicular epithelium are now made of cuboidal, and we all know what cuboidal you're taking histology right now. But instead of flat cuboidal cells, they look like a cube, right? Columnar cells. When you say simple, that means a single layer. So if I don't say anything, you understand. It's assumed it's a single layer. Columnar cells, squamous cells, like the cells on top of your skin. Stratum, the top layer is made up of a good example of that. Although the skin is stratified squamous. I won't go down that rabbit hole, but we'll talk about that when the time comes. Anyway, so they morph into cuboidal cells, and that histological change, according to some authors, marks the start point for the primary follicle. We know these guys are in the race now. Are we good with that? Nothing changes to the oocyte inside, though. It's still stuck in in fact, it's going to be stuck in prophase until 24 hours before the champion gets to release. Only the champion gets to move out of prophase 1 of meiosis. The other ones will die and wither, stuck in prophase 1. The champion goes through meiosis 1 and gets into meiosis 2, and then, I'm not going to spoil it for you. Yeah, I will. It gets stuck again, but we'll see how that works. All right, here's the picture. Okay, so here's a primary follicle. And so, here it is. And now we don't have flat cells. We have cuboidal cells. So these follicular cells are cuboidal. We have some stromal cells still kind of hanging around in the ovary tissue. But we'll see what happens to them in a little bit. But there's your primary follicle. The next thing that's going to happen, now that's arguably because some authors say another layer forms called the zona pellucida, which is super important. That guy's going to form, and some say the follicular cuboidal cells form first. Some say the zona pellucida forms first. We're going with board books, so this one hasn't formed, but that's what we're going to talk about next. This inner layer here is called the zona pellucida. All right, so next, the racing primary follicles develop another layer. And this one is right around the oocyte. Here's the primary oocyte, stuck in prophase 1, right in here. There's its nucleus. Where's it stuck in prophase 1? Right inside here where the genetic material is, inside the nucleus. But anyway, so now we have another layer, this red layer that is just formed. Maybe they formed together with these cuboidal layers. Maybe they, who knows for sure. But the way our story is going, this one forms next. This is called the zona pellucida, and we've already talked a tiny bit about the zona pellucida in the last lecture, right? It's got some ZP3 protein on it that binds the sperm head, right? Remember that from last lecture? So what is the zona pellucida made of? Well, the oocyte and follicular cells express. What does that mean, express, by the way? Express means they crank out through the central dogma. So that means a gene is turned on. Remember, RNA polymerase comes in and makes an RA transcript that swims out of the nucleus, that grabs by a ribosome that message, and it's turned into a protein. In this case, ZP1, ZP2, ZP3. Look at the amoeba central dogma if you're shaky on that stuff. I assume you know what the central dogma is. That's high school stuff, right? So the oocyte and the follicular cells, some authors say just the oocyte, some say the follicular cells help with the oocyte. Carlson says both, so we'll go with Carlson. These two cells, they turn on genes and they start cranking out some glycoproteins. Who are the glycoproteins? ZP1, ZP2, and ZP3, right? So you could also say these are the gene products. Right? These are the gene products that make up the zona pellucida. There's some other collagen, other molecules. We're not going to go into those. Of these, notice this one's red. ZP3 is super important. Why? Because this one is the one that binds to the SED1 receptors. Remember that from the last lecture? SED1 is a receptor on the head of the sperm that hooks up and binds to zona pellucida 3. Right? Remember that from the last lecture. All right. ZP2, zona pellucida protein 2, also has a secondary binding role, but it's not nearly as important as ZP3. Several authors have confirmed that. Right. Oh, here's a flashback. So Michael J. Fox in that great movie, Back to the Future. So let's zoom back, and here's the picture I had in the last lecture of the SED1 receptor. So here's the sperm head right here. Here's the SED1 receptor, and it's bound to this ZP3 protein here in blue. There's other receptors as well we're not going to talk about because the board books don't. So GALT-T is another receptor that can bind to ZP3 proteins. So it's not the only one. It doesn't even show ZP2 being bound at all. But yeah, so that's SED1, ZP3. You got to know those for boards and for my test. Communication. So there is some communication between the follicular cells and the primary oocytes. Now we have a zona pellucida. Let's go look at our picture. So how can these cells talk to the oocyte? How can our follicular cells talk when the zona pellucida is in the way? Well, there's some mechanisms where there is communication between these. Some wires stick right through the zona pellucida, and we can communicate through those. Specifically, these are called transzonal projections. Transzonal projections that project right through the zona pellucida and sink into the oocyte. So ATZP, there's two types of them, ATZP and MTZP. ATZP connects to the microvilli of the oocyte plasma membrane. We didn't talk about that yet, but I guess I should. So here's our primary oocyte. Here, there's the nucleus. So just like the enterocytes in the intestine, there are these finger-like projections. We'll see pictures of this probably next week. But there's these finger-like projections, and those are called microvilli. Okay, that's what that is. And so the ATPZ, they use gap junctions. Let's make these red. Sorry for the noises. We got, everybody's locked down, right? So we got kids running wild out there, so I apologize. There's a gap junction right there. And it communicates with those other follicular cells, the cuboidal follicular cells. Because I didn't draw that very good. But because the cell membrane actually goes around here, so they can communicate right through this space right there, right through these microvilli. Right? Because the cell membrane, I should have erased this. Because really the cell membrane goes like that. right? It's open here, so it connects through gap junctions. Anyway, so it uses these gap junctions. MTZP, there's some hormones that can go through there. Paracrine signals can go through there. Won't talk too much more about that for now. Actually, I don't think we're going to need to know anything more about that. So folliculogenesis, let's get some stuff growing. Now, Larsen is the only one who uses this term, but it's kind of neat because the follicle does grow. Most authors just call it oogenesis, but Larsen does. I kind of like that word. It is the process of growing the follicle to the point, the 50 of them are racing. One of them is going to get so big that it binds to the ovary, the outer membrane of the ovary, the tunica albuginea, and it actually ruptures through that outer membrane, and that one wins. But you got to grow to be the champion. After the zona pellucida forms, okay, there's the third thing that happens. So we got cuboidal cells formed, and then we got a zona pellucida formed. But now we are going to, that single follicular layer of cuboidal cells is going to start multiplying and multiplying and multiplying, right? So again, amazing drawing abilities. Another example of why I don't draw. But we have this cuboidal layer, right? So I won't draw them all. Zona pellucida would be underneath here. But these guys are going to start multiplying through mitosis. And another one, and another one. And this layer is going to start getting thicker and thicker and thicker. And that is folliculogenesis. We're going to grow the follicle. And again, remember the follicle is this outer layer of cells and the oocyte inside. So it's going to grow and grow and grow through growing the follicle cells. Right, so they begun to proliferate, and that's what I just drew. They become one layer, then two layers, then three. And it's not perfectly even. It starts to, some go faster than others, so it kind of starts to get messy. Once the follicle contains multiple follicles, it changes, it is called a primary follicle. Right, so that's another interpretation. I think, didn't we already make a primary follicle? We went from a primordial to a follicle, so I still let this in here. So Larsen says you have to have multiple layers to be called a primordial follicle. I probably should have taken that out because we're going with one layer of cuboidal. Other authors say it just takes that change from squamous to cuboidal cells. So I'm going to write myself a note right here to take that out, slide 77. But just to show you how authors are all over the place. So that's another definition of primary follicle. All right, so the follicle grows bigger and bigger and bigger. More follicle cells are born through mitosis, as I described. Once it starts cranking out and there starts to get multiple follicles, here's a name change we can all agree on. Larsen, Ross, Carlson. Once you get more than one layer, you call the follicle a growing follicle. A growing follicle. Growing follicles have two types. One's a primary, one's a secondary. So, I mean, really, once a primary follicle started, you have yourself a growing follicle. And remember, the rest of the 30,000 primary oocytes that are contained by follicular cells that are in the shape of a primordial follicle, they're not playing the game. They're just watching this lecture. They're sitting in the ovary tissue, just hanging out. There's the ovary. They're just like hanging out around here, where we got our follicles starting to get bigger here. Our 50 are racing. A few of them are starting to get bigger than the other ones. You can't really even see yet. But there's our race, and some of the 50 you can't even see. Here's our race trying to go here. One of them is going to get gigantic, and it's going to let the oocyte squirt right outside. It's going to be grabbed by the fimbria. There, I just did the whole lecture. But that's where we're headed. We're headed in this direction. Just remember, all these bystanders, 30,000 are still watching this race. They weren't called up to develop, so they're still considered primordial follicles. All right. The count So this is just from Ross. Now, Ross is a medical board histology book, or at least it used to be. It's my favorite histology book. The chiropractic boards use June Kira, which is not my favorite, but it's pretty good as well. So Ross goes deeper than June Kira. I like Ross better, so that's what I use. But just to point out, Ross says there's, at any time, you go back, is my drawing there? I guess my drawing didn't stick. But anytime you go back to that drawing I made, there's 3 types of follicles. There's the cheerleaders here who weren't called up, the primordial follicles, can't even see them. There's growing follicles. We're actually in a primary follicle stage, which is just a member of the growing follicle family. We haven't reached secondary yet. And then there's, at the very end of the race, maybe 2 or 3 reach the finals. Those are big Graafian follicles, or mature follicles. So those are the different types of follicles that are inside the ovary. And the flow, the evolutionary hierarchy of a primordial follicle goes primordial, growing specifically, primary, then secondary, and then to a mature follicle. So that's kind of the way we're flowing here. So we left off, we're talking about the primary follicles still. So at this point, we have a multi-layered follicular layer. So once you get a multi-layer of those cuboidal cells in there, you call that layer something. You don't really call it follicular layer anymore. That's usually for 1 layer, maybe 2 layers. Once you get 5, 6 layers in there, there's a name change. So you call that the membrana granulosa. The membrana granulosa. What is it? It's just a bunch of follicular layers that have been building up. Or you could call it the stratum granulosum. Stratum granulosum. June Kira, histology book, agrees with us here. So at this point, the follicular cells are called the granulosa cells. We said that. The granulosa cells make up the membrana granulosa. We'll see a picture in a second. Note, some authors, like Carlson, uses follicular cells and granulosa cells, AKAs. He calls the single layer of cells granulosa cells. So again, the author confusion drives you crazy. Mr. Spock's upset. He's surprised, right? He's not supposed to show emotion, so this must really be amazing revolution for him. So author problem. Ross says that the zona pellucida forms, I said this already, after the proliferation of the follicle cells. Larsen says the zona pellucida has already been formed before the word primary follicle is used. So I think we already talked about that. I guess I could probably take that slide out there. All right, so something new is going to happen. Oh, well, before something new happens, let's look now. So we saw our primary before. Let's ignore this outer layer. But now we have this thing. And look at all the granulosa cells, right? Or follicular cells if you want. Remember we had 1 layer before? There's the zona pellucida in purple. That hasn't changed. But now instead of 1 layer, it looks like what? We have 5, 6, 7 layers of cells. So collectively, these are called granulosa cells. You could still call them follicular cells. But the layer itself is the stratum granulosum. Stratum granulosum. Or that other one, that membrana granulosum, I've seen that. So stratum granulosum, I mean, that's starting to look like one of the layers of the epidermis, right? So it's probably better to call this membrana granulosa to avoid that confusion. All right. So yeah, so stratum gets really thick, but then something different happens. And there, by the way, there's that lamina propria. That remember I drew that. It used to be hanging out right here when I drew 1 layer of cells. They sit on something. They sit on a basement membrane or lamina propria. So this has been pushed way out to here as these cells have grown. So there's the lamina propria or basement membrane still hanging out there. But now we have a new layer of cells that has formed out here. We can see some new stuff out here, right? So what is that? So that is a new layer of cells as our thing, I think, gets to once there's about 10 layers, this starts to form. And that's called the theca folliculi. Theca folliculi layer. Oh, here it is right here. I even circled it in PowerPoint. Theca folliculi is now there. So we have a pretty developed primary follicle at this point. So theca folliculi, so there's a theca interna, which would be right here, and a theca externa, which would be out here. Actually, I think I have that coming again. So we have two layers. The theca interna means closest to that basement membrane. A lot of blood vessels in here. The cells secrete angiogenesis factors, which cause angiogenesis. So we're building blood vessels that can supply this growing creature. There's collagen and fibroblasts too. Some of the cells in this, the granulosa cells, have ability to produce steroids like estrogen and its own LH, luteinizing hormone. And yeah, some of these cells have important luteinizing receptors as well. Some have FSH receptors as well. Or actually, no, those are inside the granulosa cell. These are the interthecal, these are thecal cells. So these don't have FSH receptors, but they do have LH receptors. The theca externa, that was here, so it looks a lot more fibrous, right? Although you don't really, I guess these are blood vessels here, here, and here. So this is a more fibrous layer, but there's actually smooth muscle here. This smooth muscle, as we'll see, is going to help us squeeze the oocyte out of its little capsule, out of its follicle. We're going to eject that oocyte and leave the follicle behind. All right, oh, here it is. Okay, so here's the parts again. So here's the theca interna layer right here. You can see the blood vessels. And then the theca externa is out here. Primary oocyte still stuck in what phase? Prophase 1 of meiosis. It's still stuck. The chromosomes are stuck in there. Then this layer is the zona pellucida. All right, and then deeper outside of the zona pellucida, now we have the thecal layer, specifically the theca interna and the theca externa. Okay, awesome. But now something new has happened. So once we get, I said about 10 layers in this theca forms. Once we get about 12 layers deep, right? So 12 layers. Let's see if it's true. 1, 2, there's 3, 4. I don't think it's true. 5, 6, 7 layers in the cartoon. I mean, they can't keep going. But this would go out to 12. Once it gets about 12 layers deep, we start to get these little cavities forming. There's one here maybe, and one here starting to form, one here. But in one region, there's a whole bunch of these little fluid-filled cysts, because the cavity doesn't form yet. One region, a whole bunch of these cysts start forming, but other ones are forming, because this cavity is going to get really big here in a little bit. And so the next thing that happens, or before we do that, the next thing that happens is these cysts start coalescing together into bigger and bigger cavities. So now they're looking like this because they're coalescing. And then finally, they look like this giant thing here. And that has a name, and that's called the antrum. The antrum. Okay, so once the antrum can be seen in one of these follicles, these primary follicles, we get another name change. The primary follicle is no more, secondary follicle is in. And they name it a strong AKA because the antrum is what creates it. It's called the antral follicle. I doubt you're going to see antral follicle on boards because that would be too easy, because you'll know, oh yeah, the appearance of the antrum. They're going to call it a secondary follicle most likely. So again, some authors disagree. I'm letting the board books kind of guide me here. But with our story, secondary follicle is born, primary follicle is out. Maybe not at this point, maybe 15 of our racing, of the 50 follicles that started the race, maybe 15 of them, maybe 10 of them have gotten developed to the point where they have a follicle. The other ones are falling way behind. There's probably 1, 2, or 3 of them that are even past this stage. But that's how the race is kind of going. The antrum is filled with stuff. It's kind of like the amniotic cavity, right? It's filled with an amniotic fluid. This is filled with a fluid. And the fluid is super rich in a very water-hungry molecule called hyaluronic acid, or hyaluronin is an AKA right there, hyaluronin. And so all those little blue things, those are water molecules. They have bound to this glycosaminoglycan. What type of glycosaminoglycan is this and can be found in the follicle? Hyaluronic acid rich. There might be some other chondroitin sulfate in there probably, but the hyaluronic acid is the star. I don't know that. I should just say hyaluronic acid, but because the books just talked about that, so I assume that's what's in there. We'll talk about that more when we talk about the nucleus pulposus of the disc. We'll come back to these gags. Okay, but the fluid has a name, and it's called either follicular fluid, which they're not going to use on a board. That's too easy. They're going to use liquor folliculi. Is if I was writing a board question, that's what I would write, liquor folliculi. It's in Juan Chiera. That's a chiropractic board book. Even Carlson, he's the king. He confirms it. So good enough for Carlson's good enough for me. So liquor folliculi is the fluid that's inside the antrum of the now secondary follicle. Okay, and so what happens next? This darn antral, almost like the chorionic cavity and the amniotic cavity, but more specifically, when we talk about the chorionic cavity, this is the same story. So as the secondary follicle grows bigger and bigger and bigger, the antrum soon encircles almost the entire primary oocyte. Remember, just because we have a second follicle, we still have that same old primary oocyte that's stuck in what phase? No? Good. Got it. Prophase 1 of meiosis. So the antrum starts to pinch off a little stalk, almost like what's going to form the umbilical cord. There's a connecting stalk, but this one's called a hillock, and there's a little confusion on this as well. I'll show you a picture in a minute. A lot of confusion here. According to Carlson, though, the antrum really has split the zona glomerulosa into 2 layers, and therefore we have 2 new layers. The cumulus cell layer and then the mural granulosa cell layer are the 2 new layers. Let's look at a picture of this. So remember our antrum before? Our antrum was just this little space here. But remember there was other little cysts forming everywhere? And at this stage, that thing has grown huge. And in fact, here's the oocyte right here. It's almost got the thing completely pinched off. But there is a little stalk here. It looks just like the connecting stalk that holds the bi- and trilaminar disc, which we'll get to. So the antrum has split our granulosa cells into 2 layers, and therefore they get 2 names. So these ones on the outside, right here, all of these, those are called mural granulosa cells. Mural granulosa cells. The ones that are, this is where the confusion in the authors comes in. These guys are called cumulus ophorus cells, cumulus ophorus layers. But the books keep talking about a mound or a hillock that starts to develop here. Where I'm with Carlson on this. I think this cavity just kind of pushed everything apart. This is just granulosa cells left over. But nevertheless, some authors say the cumulus ophorus is just this hillock, and they use the word hillock, so they want you to use that. But this is what they're talking about. Other authors, like Carlson, say all of this tissue that I highlighted in blue, all of this tissue is the cumulus ophorus, and that's what we're going to go through. When this oocyte is, let's say this is the champion who wins the race, when this one gets kicked out to the outside of the ovary, it doesn't take everything with it. It only takes this with it. Right? It only takes this part of the cumulus ophorus with it. In fact, that changes names to the corona radiata, which we will talk about. The sperm's got to get through that. So the oocyte, when it's ejected, it'll be a secondary oocyte, but it takes these cumulus ophorus cells with it. They change names to the corona radiata. Why they don't call them the corona radiata now to save this confusion is beyond me. You'd think they would. but this is definitely an area of confusion. None of the authors are all over the place on this. But for our purposes, everything in green, we're not going to say this is the corona radiata until after ovulation occurs. Okay, everybody good with that? All right, so only one will ovulate. So again, just to remind you, we have a foot race going on, or a growth race. We had 50 or so primary follicles that were, or primordial follicles that were morphed into primary follicles to start the race. There's only going to be 3 left when you get to the end of the secondary follicle stage here. And those are the most developed. And of those 3, only one is going to be the most developed of all, and that one is going to win the race. So how do the big 3 survive and the others not? And maybe there's only one that survives and it kills all the other ones, but how do a couple of them survive and all those other 50, those 47, they die? Well, we said that this whole growth process is FSH dependent, right? So they're all growing and growing. But pretty soon, these big ones, they get so big that they secrete their own hormones. They don't need FSH any around anymore. So what a great way to get rid of the pack that's following them. You know, if you're a track and field fan, you know some runners, a little pack of 3, 4, or 5 will be out, but the big pack of 15 will be in the back. How do you get rid of that pack? You secrete something called inhibin. So once 1, 2, or 3 get big enough where they're self-sufficient on their own hormones, they secrete, genes get turned on, and they make something called inhibin. Inhibin is a hormone. It gets in the bloodstream, it goes all the way up into the brain, it hits the hypothalamus, and it hits the anterior pituitary, and it turns off the gonadotropin machinery. And so FSH is no longer made. LSH or LH is not made either. And without these hormones, the smaller ones are still, they can't make their own FSH or make their own hormones. So without pituitary FSH, they're going to die. So the younger ones, which are still FSH dependent, will die. I can see my test question playing around with these words, dependent and independent. So watch that closely, read that carefully. Don't let your eyes trick yourself. Am I saying FSH independent or dependent? I like little tricks like that. So make sure you pay attention to that. Okay, so pretty sneaky. But now we've got, for our little story, we only got 3 big ones in the race. In fact, I think we whittled it down to 1 now. We have 1 big one. Once the largest follicle reaches 10 millimeters in size, that's probably, I'll take a guess, 16, 18 cells deep, it wins. And that one's going to be called the mature follicle. They're not going to say that on boards or in my class. They'll call it a Graafian follicle. They're not a tertiary follicle. Carlson was the only one that used that term that I could find, except in PubMed they use that. But I think Graafian follicle, I should have made that the main one. So the Graafian follicle, that's the champion. The race is almost over, but now there's another race now. He's beaten the 49 other contestants, but now he's got to ovulate. He's got to get out of the ovary. All the other ones are sitting on the sidelines rooting it on because all of them are imprisoned in the ovary. This guy wants to get out. So a 24-hour clock starts once he's declared the winner. And soon this guy is going to hopefully rupture and release its load. So how does this start? So around ovulation, of course, if you have a normal menstrual cycle, 28 days, you take the halfway point, which is day 14. If you have a longer cycle, you take that halfway point. That's where your ovulation starts, right? And then you're super fertile right at that time. You can take your temperature. I think your temperature goes up. And this is not sex ed class, so I'll let other teachers. There is a class for OBGYN. Maybe they'll get into that there. But anyway, so about day 14, the menstrual cycle, there is a giant surge in gonadotropins. So gonadotropin stimulating hormone is released like crazy. FSH and LH are cranked out like crazy. Okay, Larsen calls this the ovulatory surge that occurs. All right, So this surge has some effects on our champion, and it causes the primary oocyte. So these molecules go in, there's receptors on here, and it's luteinizing hormone is the king here. Specifically, luteinizing hormone is able to get in to the oocyte, the primary oocyte, and it goes into the nucleus, and we'll see what happens here. It turns back on meiosis, and we finish meiosis 1. Let me say that again. We finally finish meiosis 1, which is awesome. So luteinizing hormone, so these cumulus cells are the ones who kind of start this, get a little deeper into how this works. Because remember, the oocyte doesn't have LH receptors. So how is that going to work if LH is the key, but we don't have receptors? The cumulus cells shut down gap junctions. This is how this all works. So the cumulus cells, they shut down the gap junctions. We talked about that was the communication into the oocyte. And we said those gap junctions were feeding cyclic GMP, some cyclic AMP. They're feeding that into the oocyte, and it was getting to the nucleus, and it was suspending meiosis. Well, the cumulus cells turn on genes which shut down those channels. And now no more cyclic GMP is being pumped inside the nucleus. So guess what? The sleepy meiosis, the sleepy oocyte that was stuck in prophase 1 wakes up and it takes right off. It goes through metaphase 1. It goes through anaphase 1. It goes through telophase 1. It goes through cytokinesis, and boom, meiosis 1 is over. And now we enter into meiosis 2. So that's the deal. Remember what meiosis 1 creates. Go watch the Amoeba Sisters. Now, I assume you know that meiosis 1 is a reduction division. So it makes kind of a clone in a way of itself, a secondary oocyte. Although, remember, the primary oocyte is what with regard to chromosome content? It's 2N. There's 46 chromosomes in the primary oocyte, right? But meiosis 1, which we finally finished, is a reduction division. So each one of these gets half of the number of chromosomes. So the secondary oocyte is one thing that's formed. It's splitting in half, primary oocyte. What are the two things that are formed? Secondary oocyte, which is 1 in 23 chromosomes. And then we have this weird polar body, which also has the other 23 chromosomes. Polar body got the short change, though, right? I'm not going to go crazy into this, but when meiosis occurred, unlike spermatogenesis, the secondary oocyte stole all the cytoplasm, and the poor other cell, called the polar body, didn't get any cytoplasm, so it can't live, and it dies and degenerates away. So remember again, meiosis 1 is a reduction division. That's a softball board question, any first quarter, second quarter question at medical school, any type of school. Therefore, secondary oocyte and the first polar body are 1N. They're called haploid. Each has 23 chromosomes. So we're good with that stuff. But now, darn it all, we got stuck again. So this time it gets through interphase 1 or interphase 2, no problem. But now it gets stuck in the next phase, which is prophase. So it gets stuck in prophase 2 of meiosis 2. I think it's just meiosis. You don't have to say meiosis 2. Prophase 2 means you're understood that you're in meiosis. So it's stuck in meiosis 2 this time, and it's going to stay that way. And the only chance it has from finishing meiosis is if it gets fertilized by a sperm. We'll talk about that next lecture. All right, so great. So now inside our follicle, we have a polar body. We have a degenerating polar body, but we have a secondary oocyte stuck in metaphase 2. We still have a follicle thick with granulosa cells. It has an outer membrane around it. And what happens now it's time to hatch this thing. So we've made the secondary oocyte. We need to get it inside the fallopian tubes. So on day 14, the hillock of the cumulus ovorus breaks down. Remember what that thing was? Here's the hillock. So this thing gets destroyed. And now look, so now we got this thing with its future corona radiata. Now it's stuck in metaphase 2, but it's loose. It can like float around in here because it's got all this space. So that's awesome. But now we got to squirt it out. And yeah, let's hatch. So that hillock breaks loose. Secondary oocyte is now on the loose in the antral cavity, floating around. And over the next few hours, the cells around the oocyte, the cumula cells, they start secreting hyaluronic acid. They crank this stuff out, and it starts grabbing water. Plenty of water is always around and coming into this thing. And this really makes the follicle get huge. Doesn't change the size at all of the oocyte, but the follicle, it's like getting ready to explode. It's so big because that hyaluronic acid grabs water like crazy. And because, everything I said, water abides and a massive expansion occurs in the intercellular spaces. And now we're up to 2 centimeters in size. Our follicle is almost an inch big. I mean, we can easily see it with our naked eye. And in fact, it's so big, it's starting to bulge out the ovarian wall and the follicle as well. And at this point, Larsen calls it a cumulus expansion. He's the only one who calls it that, so probably won't be on chiropractic boards. But we still can't hatch yet. It's ready to burst, but we still can't hatch. So we need to wither away that wall between the ovary and a little bit wall of the follicle as well. So how do we do that? We need to start a fire. And an inflammation process starts by the theca folliculi start releasing the blood vessels of the theca folliculi start releasing some well-known pro-inflammatory molecules. And that I won't get into all that down the inflammatory cascade, but an inflammation starts. And let me write something down real quick. Inflammation starts. So now this thing is ripping through the ovarian wall. It's ready to pop out. In fact, at this point, the wall gets so thin, you can actually see it through the outer layer of the ovary. And here it is. Here's an ovary right here. This is the tunica albuginea or albuginea. That's the outer layer. And you can actually see a bump right here that's pushing through that layer. And the oocyte is inside here, but that has a name. When you can visibly see it, you know ovulation is about to occur. That's called the follicular stigmata, or just the stigmata. That means within hours, the oocyte, secondary oocyte, is going to pop. It's not going to shoot out like you're getting shot out of a cannon or something. It's just going to creep out of there and kind of hang out, hopefully, on the surface of the ovary. Sometimes it gets loose, though, right? And it causes an ectopic pregnancy, but that's super rare. It's going to kind of hang out there. So it's a slow process. It doesn't shoot out. The theca externa is the last little push to get it out, though. So there's 2 more actions we have going backwards. We haven't got it out yet. So that smooth muscle that is in the theca externa, that actually contracts and helps squeeze out the oocyte. And then if the inflammation didn't do the trick, there are some collagen-degrading enzymes we won't worry about that are secreted from the theca externa cells, and that does the trick, and it rips a hole through the ovary, the wall of the ovary. And now the secondary oocyte is outside of the ovary. Okay, down the hatch. So here's the fallopian tube, right? This is the fallopian tube. Remember, there's the ampulla. The isthmus would be connecting to the uterus. It'd be up in here somewhere. Then we have the infundibulum, is this region right here. So this is all ampulla from here up to the infundibulum, right? This is where fertilization occurs in the ampulla. But the ovary, the little egg, was sitting on the ovary right here. And so these weird little fingers that are way up here, somehow it knows, and I'm sure molecular biologists could tell you how. I'm not going to go down that rabbit hole. But these things know, and they actually move down, and they cover the oocyte, the secondary oocyte, and then there's little cilia on these fimbriae. The fimbriae don't move themselves. They don't like sweep it in. But there's microvilli. If I blow one of these things up, these little fingers, there's tons of little hairs that called not microvilli. They look like microvilli, but they're not. They're cilia. They move, and they sweep. They'll grab that oocyte, draw the oocyte, and they'll start to sweep it right down the tube, and that's what happens. And once it gets in the tube, we'll talk about the sperm. These guys are going to meet up, and fertilization is going to occur right there in the center of the ampulla, hopefully. All right, we're all good with that. Okay, so that's the end of that story, but we got a little cleanup to do because there's another important thing. That giant tertiary follicle that gave rise to the oocyte and accomplished the winner, the ovulation was accomplished. What happens to that thing? What about all those granulosa cells? Does it go to waste? Well, not initially. The granulosa cells, they will divide by mitosis into a new cell type called luteal cells. And these luteal cells start to proliferate, and you start to get a structure again. It starts to kind of beef up. After the follicle leaves, you have kind of a collapsed bag. But now it starts to thicken up because you have a new cell population called luteal cells. They'll repopulate the broken down follicle, and therefore its name changes. No longer a tertiary or a Graafian follicle. It changes to the corpus luteum. And corpus luteum is an endocrine gland. It is an endocrine structure, albeit temporary, because it secretes a bunch of hormones. Talk about that more in fifth quarter if we get that far in endocrinology. Who is this guy? So what do these hormones do? The hormones mainly are estrogen and progesterone. And those guys get into the bloodstream, and they get into the lining and the meat, or mainly the lining of the uterus. And they get it ready for fertilization to occur. Assuming if an egg gets fertilized, it needs to stick to the wall of the uterus. I don't think I put this in here, but here's a uterus. Here's a fallopian tube right here. Okay, there's the ampulla would be right here. Okay, there's the ovary where it was connected. And so we got fertilization occurred here. I guess I can make it pink. Fertilization occurred here. And now we got this fertilized oocyte coming down. It turns into a morula, which we'll talk about. It goes kerplunk in the uterine cavity, which is filled with a juice. Its job is to stick to the wall of the uterus, and then it actually goes inside the uterus, and that's where it grows, and it grows, and it grows, and it grows, and a baby starts to develop right in here as it grows. All right, so that's kind of where we're going. Might as well draw this more. There's the cervix down here. Yeah. So that's kind of where we're going. Now, of course, we're going to go over this more in detail. But this has to be prepared for such an event to occur. There's got to be extra blood vessels in here. So tons of blood vessels start developing here in the endometrium, getting ready for this implantation because it has no circulatory system. It's got to feed it with blood. And so that gets really thick. If fertilization does occur in here, tons of blood vessels grow, and we're off to the races. If it doesn't, this lining it sloughs off, and that's what the woman bleeds. That's the end of the menstrual cycle, and that's called your period, right? And that's kind of how that works. But who prepares it? The estrogen and progesterone are needed to prepare the endometrial lining, and that is coming from that dead tertiary follicle, which is turned into the corpus luteum. So very cool. All right. Yeah, so luteal progesterone, meaning luteal, meaning it came from the corpus luteum, stimulates the endometrium to thicken and become vascular in preparation for implantation. If it fails, corpus luteum quickly degenerates into a scar tissue structure, and that's called the corpus albicans. That's the dead corpus luteum. That's the dead tertiary follicle. All right, I think, are we done? Yeah, of course, I don't know where my iPhone went, so I don't even know what time it is, but this is some pretty heavy material. Should we go a little further or should we stop? Let me see if I'm trying to see how much time I have. That's probably a lot to digest. So, the next video we will do Meet the Ova. That we'll actually go through the process of fertilization is where we're going next. All right, so I will see you next week. Again, as always, if you have any questions, send me an email. Be glad to help you. This is not easy stuff, but hopefully I made it so you can understand it. I will post these slides on Brightspace, so you'll have them. Definitely you want to download those and use those to follow along when this is started. All right, see you in the next video.